Hi guys, my name is Brianna Scott, and I'm going to be presenting to you today on the topic of bats and how they are hosts for diseases, but have antiviral protection against those diseases. So, as you can see, I titled this 2020 the Year of the Bat for obvious reasons. Going through this year of COVID and the changes this pandemic has brought about really drove me to dig into this topic, saying that initially when I first found out about COVID, it was said, it was reported that this all occurred because of bats. It began with bats and it was now spreading to the human race. And now here we are, 8+ months into this pandemic, or right at 8 months into the pandemic. And I thought it would be really interesting to understand what was occurring that, um, bats can pass this along to us, but they're not necessarily dying from it. My initial topic was not this. My initial topic was on plant immunology, and I just could not wrap my head around what was occurring with the plants. So I wanted to grab something that was a little more relatable. And this is what I came to. This is what I found when I was digging and doing my research, looking for primary literatures that I could use. And yeah, so this is what we got. So we're going to be looking at positive selection of serine residue in the bat, which confers, IRF3 confers enhanced antiviral protection. And I know that's a lot, but we're going to unpack it and really get down to what it is that these scientists suggest uh, could possibly be leading to the reasoning behind bats not getting sick, even though they host many, many viruses. So just to give you the rundown, we're going to go over um, background and then the IRF3 pathway, some key terms that we might want to remember, what the scientists hypothesized, the methods, results, and then just some key takeaways from this study. So here we start off with a simple major lineage tree, just showing the two suborders. So we have the yin bats and the yang bats. And by all means, if you are able to pronounce these words without fumbling, go right ahead, but I'm not, so we're just going to keep it simple. And when I say yin, I'm referring to these bats, and then when I say yang, I'm referring to these bats. So there's well over probably 1,400 species of bats, and we are more familiar with the yang bats. So these guys down here. But I bring this up because the yin and the yang bats are the two suborders that they actually they pick several bats to test from each suborder. So that's why I bring that up. So diving into a little more about the background of bats. Bats are really interesting. It was brought to my attention that bats are hosts for zoonotic diseases. I never knew that until now. Until the year of COVID, until the year of the bat, I did not understand that bats had the capability of carrying up to maybe two viruses sometimes at a time, and really how complex their small bodies are, because they carry many zoonotic diseases. They can carry I think I found that 60 plus viruses, zoonotic diseases, have been found within bats alone. That's not including other mammals or animals in general. So, zoonotic diseases are diseases that can be passed on from an animal to a human. This means that there may be some disease, and not to scare anyone, that your dog could have that could be passed on to you, or if you have a farm, your cows could have that could be passed on to you as a human. Bats also have that capability of hosting zoonotic diseases, and they jump from bats to humans. That's how it's thought that COVID became what it is now in this huge pandemic. The interesting thing is that bats harbor these zoonotic diseases, but are asymptomatic. They don't show any symptoms of having Ebola or having rabies or having COVID. They don't show any symptoms when it comes to harboring these diseases. They can simply transmit them, transfer them, pass them on, but they don't get sick. Excuse me. So the question that we're here to answer is, what's the difference between a human and a bat? Why do bats not get sick when they have COVID? Why do humans get sick when they have COVID? And there are some humans that are asymptomatic, but the the real question is, what is causing this? And so that's what we're going to dive into today, and what my paper was on. So just to introduce you to a few things that occur within the paper, this is called the IRF3 pathway. And essentially what we're looking at here is an interesting experimental method, an interesting experimental system, let me say. So what they did was they introduced a protein called poly-IC into the system. Just think of a body. And the poly-IC mimics a protein that a virus would put out. So this is a double-stranded RNA that mimics a virus. There's no virus involved, so it's not actually going to make whatever you're infecting sick, but it's going to turn on the immune response as if there is a virus present. So the poly-IC enters, and when it enters, it's going to turn on some sort of gene, whether it be RIGI, MDA5, TLR3. In this case, we can just say it's turning it on MDA5. And when that's turned on in bats, IRF3 is initiated. This has a serine at the 185 position, and it creates an antiviral response, a huge antiviral response. So bats aren't going to get sick because once that poly-IC turned on the immune system, that gene activated the IRF3 antiviral response for bats. And that's because, like here in red, you can see at the serine at 185 on the IRF3 allows for that antiviral response to occur. Now, when the poly-IC, this double-stranded RNA that mimics a virus, turns on the gene in humans, the IRF3 is activated, but leucine is at the 185 position. And you have a very minimal antiviral response. So compared to the bat, who has serine at 185, the humans have leucine at 185. They get sick. There's little to no antiviral response. Their body can't protect them from this virus. But the bats, on the other hand, have a huge antiviral response, and they don't get sick. They're asymptomatic. They're flying around, having a good time. So, and maybe just to help us remember, red, serine at 185, bats don't get sick. Green, leucine, 185, humans do get sick. So just a little brief overview of how that pathway works. A a few key terms. IRF3, which is an interferon regulatory factor. This is the factor that we're, really honing in on and trying to decide whether that leucine or that serine that's on there really does make a difference. And just from that brief introduction we just did, we can kind of see that it does, but we'll dive into it a little more. And serine residue, this is in the title of my paper, and I just thought that it would be good for you all to understand that a serine residue means a serine amino acid just in a specific location. So S185 is our serine residue. And then as far as poly-IC, just remembering that it is not actually a virus, but it mimics a virus. And so it is going to initiate, it's going to turn on the immune response necessary for the organisms to do what they need to do as far as allowing the virus to replicate or not allowing the virus to replicate. Okay, so just again, we're going to remember serine inhibits viruses in bats. Leucine does not inhibit viruses in bats. So serine at the 185 is going to cause bats not to get sick, while leucine at 185 allows humans to get sick. So before they actually went in and did some experimentation, they just wanted to do some computational work. So what they did was they actually decided to do an alignment analysis, and we're just going to focus on a couple parts here. The circle parts, if you can see, over to the left, these are just the bat species. These were the 11 bat species, and then in the middle here, if you squint your eyes, you can see that this is the positively selected area of those sequences. So at the 185, as you can see, if you go down to the little box and line them all up, several of these bat species have an S in the 185 position. Not all of them, but most of them. I think 7 of the 11 that they were conducting the experiment on actually had positively selected for serine in that position. And when we say positively selected, that means over time, as these species have evolved, they were getting sick. They were dying from having these viruses. And over time, as they evolved, genetically, they changed. And that change ended up resulting in some of them now in the 185 having serine instead of leucine or valine or whatever the case may be. They now have that serine. And once that serine is in that 185 position, it's actually going to show that that selection is necessary in order for their survival and for them to not die from all the viruses that they're able to host. Okay, so just introducing my paper to you. The title was Positive Selection of Serine Residue in Bat IRF3 Confers Enhanced Antiviral Protection. And hopefully this kind of makes a little more sense now that we've gone through a little bit of background and such, but let's get into it. So the authors of this paper, the scientists behind all of this work, we hypothesize that coexistence with RNA viruses has imposed strong selective pressures on bat antiviral signaling molecules, resulting in robust antiviral responses to virus infections or immune activation signals. So they hypothesize that serine is going to cause a huge response, and leucine probably won't cause a huge immune response. So saying that the serine is going to cause the organism not to get sick, and the leucine at the 185 is probably going to allow for the organism to get sick. So in their method, what they decided to do was they took human cells and bat cells. Not just regular human and bat cells, they took human IRF3 KO cells and bat IRF3 KO cells. So what this means is that these cells were actually turned off. Their functions were turned off. KO meaning knockout, literally. So they knocked out the pathway for IRF3 to function in humans, and then they knocked out the pathway for IRF3 to function in bats. This leaves these cells defenseless. If we were to attack them with a virus at this point, the virus would replicate and they would be taken over. So now these defenseless cells were clean slates for them to work with so that they could manipulate them in a way to figure out what is actually causing the better immune response. So they entered a leucine into the human cells. They entered a serine into a human cell. And then they entered a leucine into one of the bat cells and a serine into one of the bat cells. So now we have our cells, the cell with serine and the cell with leucine. Leucine being the human original amino acid at the 185, usually allowing for low or no antiviral response, meaning that the humans get sick. And then you have your serine, which is from the bats, from the 185 position of the bats, that inhibits viruses from actually replicating and reproducing. So this cell shouldn't allow virus replication. On the other hand, we have the bats as well. They have the serine, which is the original way that they come. They don't get sick. And then they added a cell with leucine. And so the idea is that this leucine cell is going to have virus replication. But even though they now have their prospective cells how they want them, they now need, these are still defenseless cells. So if they were attacked right now, you're going to see enormous amounts of virus replication. But before we attack them, they introduced poly-IC, which mimics a virus, but not actually introducing the virus. So this poly-IC is going to rev up the immune system. It's going to go ahead and turn on the immune responses, allowing these cells to go ahead and prepare for attack as if they're being attacked by a virus. They're going to turn on their defense mechanisms. Once the poly-IC has been introduced to the cells, we then attack it with the VSV virus, and we wait. They wait to see if there's going to be virus replication or not, either in the serine cells or the leucine cells for bats and for humans. So in order to see this quantitatively, they attached a GFP, a green fluorescent protein, to the viruses that they introduced. That way they, could use the light and see the actual growth of the viruses on the plates. So across the board, the results were pretty straightforward for the yin and the yang bats and the humans. So on the x-axis, you have the VSV GFP, which is the virus that they attacked these cells with, and the growth. So this is going to show you, as we go further up, the infection. And then down below on the y-axis, it's showing us how much of the, they call them plasmids, but the serine and the leucine were introduced into the cells. So over a certain amount of time, they would introduce a little more of the serine and leucine and see how that affected as well. So in this first box, well, not box, but in this first section, there's no poly-IC. So remember we talked about poly-IC mimicking a virus and turning on the immune response? Well, in this case, there's no poly-IC. So if there's no poly-IC, these cells can't turn on their defense mechanism. So these are helpless cells, and we can see they're pretty helpless. The virus grows in all of them. It replicates in all of them, including the serine. Now, a little less in the serine than in the leucine. So serine is blue, leucine is green, but they still both got sick. We would assume that they still both got sick. So on the right-hand side over here, the poly-IC is introduced, the immune response is kicked on, it's turned on, and then leucine and serine are introduced. And when they're introduced, the immune response is greater for serine than it is for leucine. And we can see that because the growth of the VSV, of the virus, is very minimal for serine compared to what it is for leucine. And so this is just solidifying the idea that bats have positively selected for serine at the 185 because it does indeed create antiviral reactions. It does allow them to not get sick. Humans obviously have not positively selected for that just yet. We probably should get on it, but humans have not positively selected for that serine in that 185 position, and instead we have leucine, and we still get sick. So just some key takeaways. Zoonotic diseases are a real deal, real thing. I wasn't really aware of zoonotic diseases until this COVID outbreak. I never really thought about the fact that there could be a disease that can jump from an animal to a human. And definitely not in the capacity that COVID has, because who would have thought that we would be in a pandemic right now? But zoonotic diseases jumping from bats, cows, dogs, it's kind of a scary thing to think about, but just understanding that these zoonotic diseases do exist and that we should really probably start doing our research on our best ways to defend ourselves from those diseases. And just understanding that bats carry a plethora of viruses and don't get sick. That's really cool to me. I mean, I wish it were the other way around, but it's still really cool that bats have this capability. Bats are closely related to us, and it's just strange that they were able to do that and we weren't, or we haven't yet. Maybe we will, who knows. But if you were interested in this topic and wanted to dig into it a little more, I know they were saying that bats' ability to fly long distances and certain things about inflammation in their body and how they can keep down inflammation is what led to these changes. So not to get too deep, but just something for you to look into if you were interested in trying to further along your research on why it is bats don't get sick when they harbor these viruses. And then last thing to take away is IRF3 immune proteins differ between humans and bats. So this experiment suggests that serine is the better selection, the better way to have evolved. Serine at the 185 is going to cause bats not to get sick, while leucine at the human 185 is going to cause humans to get sick and not have an antiviral response. And I think it kind of put in my head this question of the humans that do contract COVID and are asymptomatic, are they more like bats? Do they have the serine at the 185 locus instead of the leucine like most normal humans do? And could that vary within families? It's odd because someone in your family could have COVID and be asymptomatic, but then someone else could get COVID in your family and be, horribly ill. So just something to think about. And I really hope you guys enjoyed this presentation. I found it really interesting, and I think I am going to do a little more digging. But just wanted to say thank you. My daughter was a little baby bat for Halloween, so I thought I'd insert her little photo here and tell you guys thank you for watching. I hope you have a great day.